All matter is made up of atoms. What are atoms? Well, they're made up of these three things, and that's as deep as we need to go. In the nucleus, we'll have our protons and neutrons in equal numbers. Protons have a positive charge. So for an atom to be really what the element that it is, we have an equal number of electrons surrounding it as what we have protons in the middle. And they're hanging out in these shells on the outside, circling in their little orbitals. And I really don't need you to get too much more specific in this. You learn that in chemistry. Congratulations, okay? And that's going to be pretty much it as far as the atoms part. We're going to talk about bonds next. But in order to understand the importance of why we're talking about bonds and everything, we get into this concept. What's a molecule versus what's a compound? Compounds are molecules, but molecule is a bigger term that covers anything that consists of more than one atom. But if it's two different kinds of atoms, two different elements in it, now we're at least two, it could be more, we're getting to compounds. Okay, that's our terms. There's three types of chemical bonds, and these are actually pretty crucial for understanding how biology functions. So our three major types of bonds that I need you to understand about are really two bonds and an attraction force, really, okay? So our two bonds that we actually have electron movement going on are covalent and ionic. In a covalent bond, we're actually sharing our electrons between two atoms. I think of this as being like when I have to share my Barbie with my sister. It's going to be a great analogy, actually, because I'm the older sister. My sister Erica, she's younger than me, 2 years. Do you think I was really like fair and shared that Barbie equally with her? Or do you think I shared it when mom was looking? Right. So I might have hogged that Barbie a little bit. We have some atoms that have a tendency to hog the electrons too, the Barbies. All right. Oxygen is, completely famous for that. We'll say, as a rule, if you see an oxygen in something, it's probably hogging the electrons for itself. And that's really important because, yeah, it is still sharing a little bit with the other things that it's connected to, but it's probably hogging it a lot more than it probably should. And so that makes it have a more negative polarity. And the things that it's attached to tend to have a positive polarity. There is one kind of molecule that it makes the basis of life, carbon, which is always going to be the fair sharer. It's always going to get its time that it wants with the electron, and anything that's bonded with it will get its fair time, and it gets to make that dictation. So think of carbon as the great equalizer in all of that. Okay, so if something's bound with carbon, and whatever it's bound to, they're sharing equally. But if oxygen is bound to a hydrogen, that hydrogen doesn't stand a chance. So oxygen's going to hog that Barbie no matter what. Okay, it has a way stronger backbone than its little sister, hydrogen. Okay. All right, so that's the idea. So sharing our electrons, that's our covalent bonds. I don't care too much about you really understand what's going on with like double and triple bonds, as long as you understand what the covalent bonds really do in there. All right, so when I start hogging that electron or hogging that Barbie, oxygen is what we call electronegative. It attracts the electrons stronger. It keeps the Barbies more likely to have that personality to keep the Barbies to itself more. And then its little sisters, they're attached to it, the hydrogens that it's supposed to be sharing with, they just don't see that electron as much. So because they're not seeing it as much, they have that positive polarity to them. This is what basically defines how all of our biological molecules are going to be behaving in your body, because you're mostly water, right? So you're talking about this hoggy oxygen attached to these piddly old hydrogens with their positive polarity and that negative oxygen. And that's going to basically dictate how everything else in your body is going to be behaving with it. Critical that we remember that that's what's going on with water. The other wonderful, beautiful thing about water is that that oxygen can get so greedy that it'll completely steal that electron from the hydrogen. We'll get into that in a moment, okay? Because that's going to bring us into ionic bonds. Ionic bonds is whenever an atom steals the electron from the other guy, or you can think of it as donating or transferring, because sometimes they're happier about it. If we look at sodium and chloride up here in this picture, sodium's got an extra quote unquote electron out here. Now, if it had, 11 electrons out there to match up with its 11 protons, well, that's fine. It matches up to make it the sodium. But that extra electron in that outside orbital, it's like, cool, like I'm alone out here. I don't really need to be here. It meets up with chlorine. Look at this guy with its 17. It's got 7 guys out here and it's got this one open blank space that would love to just fill it up to a full 8 and make it even more stable. So sodium says, I got this, Barbie. I don't need it anymore. Or even better, if we're sisters and I'm the older one, I outgrew this shirt. You can have it, man. I don't need this shirt anymore. It doesn't fit me anymore. Congratulations, Erica. You got a new shirt. That's yours now. I don't need it back. We don't need to share it. You can have it. So she's up a shirt and I'm down a shirt. And that's basically what's happening with sodium and chloride. Okay. So sodium, you can have my shirt. Chloride says, great, I needed a shirt. That's going to fill up my wardrobe. Now I have more options here. I'm a lot happier now. But that'll make sodium positive because it's got more protons than electrons now. And that'll make chlorine negative. Wow, that really committed, didn't it? Anyways, that's negative charge because of that extra electron that it's carrying compared to its protons. But those outside orbitals are going to be more stable for it. This is what happens in ionic bonds, and it makes a lot of sense when we're looking at things like sodium chloride, where we can have that exchange happening. And then when it's just salt sitting there, it's just sodium and chloride, and they're existing together with this in their ionic bond, and it's nice and happy. When we introduce it to water, though, that's going to get disrupted. And so we're going to get to in a minute. But those two, so the sodium and the chloride, when they're put in that solution with that water, with the polar oxygens and hydrogens, those sodiums and chlorides don't need to stay together anymore. Those charges can start interacting with other molecules that have charges too. That's ionization. Where now we can go separate ways. Erica can go wear her shirt in middle school, and I don't need that shirt anymore now that I'm in high school. We're going our separate ways. Okay, so think of it that way. And so that'll create cations that have our positive charges. I say cations, you can remember they're positive because the T is like a positive sign. And that's how I remember which one is which. Anions, an is kind of a negative, connotation anyway, so I think that would be negative. And then electrolytes that are able to do this, they are going to ionize in water. And these are pretty common molecules that we actually literally treat as electrolytes in our diet that help keep that balance of what we need in our own cells. We'll talk about, I know you guys are probably already thinking into, we're going to talk about hypotonic and isotonic and hypertonic, which you probably learned a little bit about that in chemistry. We'll talk about that in chapter 9. That's not for today. Okay, but we will get to it. So understanding that concept is going to come up. Okay, but here we can see here, if we put sodium and chloride in water, those nice negative chlorines that have their extra electrons because of that ionic bond leading to the ionization in the water, that negative with the positive of the hydrogens. You saw it happen, right? That wasn't me. The negative of the oxygen with that positive of the sodium. And they're happy to be associating with those polarities. Now you can think of these polarities as being almost sort of like drama. Drama is going to beget drama, right? Because later we're going to be talking about that nice, stable, strong, sharing, peaceful carbon and how we're not going to have any charges associated with the hydrogens on those carbons. And how that non-polar molecules, they are nice and stable and chill. This chaos doesn't want to have anything to do with that, does it? So that's important to remember too. Now we're going to get into hydrogen bonds. So a hydrogen bond is an attraction. When we talk about covalent bonds sharing electrons, we talk about ionic bonds donating or transferring electrons, full on moving an electron over permanently. Hydrogen bonds are just attractions. And just like the name says, there's always a hydrogen involved. So if you had a positively charged, I don't even know because I'm not that great with like inorganic chemistry stuff. If we had a positively charged, I don't even know what, a calcium that was attached to something else. And then we had a negatively charged chlorine that's attached to something. They're still attached to those things. And those attractions from the chlorine to the calcium, well, they're still attached to those things. Is that a hydrogen bond, those being attracted? No. Why? There's no hydrogen. And that seems simple enough. But remember that. It's always going to have a hydrogen somewhere in the attraction. And it's important to remember that because when you look at water, here's a water molecule, right? H2O. Water, I used to have like a little writing pad up here. And I don't know how to erase without like going back. So just pretend like that didn't just happen. This says H2O, I promise. Goes back and forth between these kind of two states. H plus and OH negative, because sometimes, like I said, really, that oxygen gets so greedy that it completely steals from that one of the hydrogens. And so when I draw the two arrows the two ways, I know you've seen it before, but that means we're kind of, it's like Schrodinger's cat in a way. You can't really say that the water is just always H2O in your water, because it's also H plus and OH negative. And yeah, if you want to get really picky with it, you could have hydronium ions that are like H3O plus, and we can get, forget it. Okay. So just focus on the H pluses for me. Okay. So the H plus and the OH negative. So if I give you pure water, and it can only ever be either H2O or H plus and OH negative, right? It can only be those things. You would have to have equal H pluses and OH negatives, right? It's not going to be outside of that. If it's just water and you're only introducing H2O, it's either going to be H2O or it's going to be H plus and OH. So you're always going to have equal H plus and OH negative. Seems like I'm bringing up a point that might not matter, except that we're fixing to talk about pH. Okay. So do you guys remember what pH scale is? 0 to what? 14. Right. I know where you're getting at though. Okay. Yeah, I know where you're coming from. This. I have to actually break out the freaking eraser. Can I left click it, do you think? No. Okay. That would be asking too much, wouldn't it? Okay. Bear with me. We'll try again. Here's a 7. That's a lot better because I need this to be like at least somewhat legible. All right. So we know 7's in the middle, and probably you guys know that 7 is neutral. But why is 7 neutral? It's literally because it's in the middle. Because your H pluses have to equal your OHs. And when your H pluses have to equal your OHs, you're equal on both sides of it, and you're right smack in the middle. That's 7. And that's where that why 7, why water is the middle. Okay. Don't ask the question for the 14, please, because I don't know why 14. And I know that there's chemists that know that, and I don't care. But for your purposes, if you know that 7 is neutral, and I'm telling you H2O is always going to be equal H plus and OH, and that's why 7 is in the middle, then what could the maximum be? It would have to be double that, 14. Okay. Now we've got our scale of 0 to 14. 7's in the middle because your H plus and your OH are equal in water. You start decreasing the ratio of H pluses, you start decreasing the ratio of H pluses, we're going to go to the base side. I'm going to talk about why that is in a moment. And start increasing your H pluses, you're going to the acid side, which I'm sure you guys remember, towards 0 is acid, right? Less than 7 is acid, and more than 7 is base. And we're going to talk about why that is in a moment. Before we get into that, we're going to focus back in on that oxygen molecule and that older sister, the Lauren in the group, if you will. The oxygen, it's me. That oxygen being electronegative and then having that negative polarity by hogging those Barbies, right? It's got negative polarity, which means we're giving those H's a plus, positive polarity. And negative likes to associate with positive, opposites attract, like the Paula Abdul song. Does anybody know what I'm talking about when I say that? Okay, thank God. I said that once, and I was like, it's like the music video with the cat, and everybody's looking at me. You're all looking at me like I'm crazy. Opposites attract, and it ain't easy. It's a natural fact. Anyways, it's from the 90s. And anyways, so opposites attract. They do. In chemistry, this is really important because the opposite of that negatively charged oxygen atom and that positive of that hydrogen that's not seeing that electron as much, they're going to be more likely to hang out with each other. It's actually why if I took a cup of water here, why that water wants to bead up on itself, because we've got all of these water negatives associated with positives, and they kind of hold each other together. Those attractions between that oxygen that has that negative charge and that hydrogen that has that positive charge, that attraction there, where we have these dotted lines, that's a hydrogen bond. That's a hydrogen bond, an attraction. And we have a hydrogen involved. We'll make sure the hydrogen bond. Okay. We clear on that? Because I swear is probably one of the most important concepts that's going to come up over and over and over and over again. Understanding that. Because when I talk to you guys about DNA, we think about our DNA being like this, right? Blah, 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 blah. Here's your rungs of your ladder. And we know these guys meet in the middle. And we know this is a double helix, and it's double because you have two sides to it, and they are matching up to each other in the middle. And you guys probably looked at this a little bit in chemistry. We're going to talk about it a lot in chapter 6. So this is what your DNA looks like if you unwind it into just a straight up ladder, right? What's holding those two sides of the ladder together? Hydrogen bonds only. The two sides of your DNA strands are held together by that kind of attraction. And it's oxygens and hydrogens. Hydrogen bonds hold your double helix together, and that's it. Tell me that's not impressive now. So just attraction is holding your DNA together. Now when I tell you, anybody know the pH, what pH your body should be? Anybody remember that? 7.35 to 7.45. And if you go to the doctor and they test your blood pH and your pH is 7.48, do they think they're going to be like, oh, it's a little off? They're going to be like, holy shit, we need to get, some acid into this guy and balance that out. This is bad. We need to figure out what's going on. Balance it first and then get to it. Why? Because if hydrogen bonds are holding your DNA together, the structure of your proteins that do everything in your cell, proteins do everything in your cell. Those structures of those molecules, which are so essential for the functioning of anything in your body, are held together by hydrogen bonds. If you have too many or too few H pluses, you get that out of whack. Now you're getting the bonding of all the molecules in your body that are depending on attraction. You're interfering with their ability to attract the way that they're supposed to. That's why it's dangerous. We're talking about every molecule pretty much in your body depending on hydrogen bonds in order to function the way that it's supposed to. You can't have that balance getting off. So now we're starting to understand a little bit better why I'm going to drive so hard with understanding hydrogen bonds and pH. Because those things are extremely important, and they're going to be just as important in physiology. If you haven't had it yet, you're all going to have it at some point, right? Probably taking it next semester or over the summer. It's coming, right? So if you haven't had it yet. All right, so blah, blah, blah with these terms. I'm really bad. I'm not going to lie. I have a bachelor's degree in biochemistry. I graduated magna cum laude. I had 149 hours in my 4 years at undergrad in biochem, okay? I still get solute and solvent confused sometimes, all right? So you're not alone, okay? So this is one of those things where I wasn't reminding you about it, but I don't talk a lot about solutes and solvents that much in my class, but hey, it's one of the things I have to touch on again in chemistry. So a solvent is the thing that we're dissolving the thing in. The solute is the thing that's getting dissolved in it. So salt would be your solute. Your solvent is typically going to be water in biology. Okay. The solution is when you have a liquid and stuff's dissolved in it. It really is that simple. And as far as concentration, that part, most of us understand concentration, right? It's how much of a thing is dissolved in a liquid. That's the basics of it, right? So yeah, we can get down to that nitty-gritty of it's how much solute is dissolved in the solvent. Yeah, it is, but like it's how much stuff is dissolved in the liquid. Okay, so keep that in mind because that's important because we are getting into pH in a moment. And does that have to do with concentration? It surely does. All right. So we talked about solutions, and it's important because look at all these little water molecules out here. These guys aren't just associating with our electrolytes like sodium chloride. They're also associating with pretty much any molecule that's going to be associated with your body. They're having some sort of interaction with, whether it is with because they're also polar or because they don't want to associate with them because they are non-polar, right? That brings us to that concept of hydrophilic, hydrophobic, and amphipathic. Thank God for some words that have the definitions in it, right? So hydrophilic, water loving, like the city of brotherly love where I went to graduate school. So hydrophilic, water loving. These are going to be molecules that have some sort of charge or polarity to them. They're down to get down with that chaos party of the charges that water is bringing, okay? Hydrophobic molecules, these guys are so stable. It's like going to a party where like everybody is like off the wall and you swear they're all manic or they're on something and everybody's crazy and you're just super chill and stable and you've got kids at home and you don't play with drugs and you don't drink and you got to keep a level life and whatever, you're the carbon, okay? And you got it all together, relatively speaking, in those moments, right? So we're nice and stable and we're not trying to play with polarity. That's where hydrophobic comes in, water fearing. Those are our non-polar molecules. Okay? If you have both aspects, you're amphipathic. And I'm sure you guys Probably have learned about like phospholipids, wire phospholipids. I'm going to show a picture of one in a little while. But wire phospholipids, amphipathic, because y'all seen a phosphate, right? These things are wild. When I'm sitting here talking to you guys about like oxygen and how greedy it is, and I can't ever remember how many bonds that phosphorus makes. I think it's 6, but and then whatever. And then this is attached to. Good. I'm glad you did that. This. Now I've drawn it like this, and you guys learn in chemistry about this is very beautiful about what each of these notches means. It means a carbon is there, and if a carbon's there, it's attached to hydrogens. They're just not drawn on here, right? We love these images. Anyways, so these might be a whole bunch of hydrocarbons. Carbons and hydrogens, happy, stable, living their hydrophobic life down here. And this is a phosphate. If you see oxygens in biochemistry, automatically just be thinking electronegative. They've probably got a negative charge to them. Phosphate sure as heck does. I mean, it is negative. This is crucial because now we've got this negative head and this non-polar tail. So we've got both going on. Hydrophilic, water-loving, hydrophobic, water-fearing, right? So that makes it amphipathic. And anytime that we're talking in my class or in any other sort of biologically based classes, if you hear amphi, always think both, just like amphibians. They're water and land. So we'll have a lot of amphi terms kind of come up. So I'm warning you ahead of time. Amphipathic in this case, going along with hydrophilic and hydrophobic aspects. Okay? We'll come back to that phosphate in a little bit. Phosphate plays a pretty big part in biology. So we're going to jump into the pH scale. I actually don't want to start off with this figure. Let's go to the actual scale itself. I'll redraw this because this equation is important too. I want to try to get into explaining this. So we kind of already introduced that concept of water being able to go back and forth between H plus and OH. And that's why it's in smack dab in the middle. If we can remember that water is pH 7, and most of us know that, then you know, and I'm telling you that it's in the middle because of that H plus and OH being equal, then we would know that it should go all the way up to 14. Okay? But pH literally, if we're looking at what that number is, it's the actual negative logarithm of the concentration of hydrogen ions. That's literally what it is, okay? It's something with the computer. It's not what I'm doing, I promise. And sometimes I just want it to look nice, and sometimes I don't care if it messes up. It's just one of those times I want it nice. Okay, so we have our nice little pH equation on here, and I'm going to break it down for you one more time. I know you learned it, but this is really going to play into a lot of stuff we're learning later on. So it's the concentration. Those brackets just mean concentration. Okay. So it's concentration of hydrogen ions, and that's how we're looking at it, okay? So, and logarithm means it's based in the scale of 10. Thank God for that too, right? I tell you what, I'm not even joking when I tell you I'm bad at math. Okay, and then this negative means it is an inverse. So let's just start with 10 in the base of 10 first, okay? So if I write this number on here, 10 to the 2, just 10 to the 2. I'm gonna lose my patience real quick with this. So that, I promise, says 10 to the 2, okay? And 10 to the 2, we know that what that really means is 10 times 10. Why does it do that? I don't even understand, because I'm not doing anything weird with clipping or anything. 10 times 10, and we know that means 100. Another way that you can think of it is with that 2 up there is how many zeros you're going to have after a 1. That's another way you can look at it, okay? So if I put 10 to the 5, as another example, that'd be 10 times 10 times 10 times 10 times 5 of them. Or you could look at it as 1 with 5 zeros afterwards. Oh my God, 2, 3, 4, 5. You could think of it as like that. So it's kind of telling you how big that number is. And we know that like 10 to the 14, I mean, you're going to have a ton of zeros after it. So that's just looking at just 10. But then if we break it down to the negative or the inverse of all that, which is what this negative means in front of the log, now we're talking about 1 over whatever that number is at the top, that exponent, right? That's right, exponent, right? I'm not saying that wrong. Okay, I don't know why suddenly it was one of those moments where you like the word comes out and you're like, Am I speaking the same language as I was earlier? Like when you say the word please enough and it starts to be like, is that the right word? Is that, I don't know. Anyways, so 1 over x. And then so that would be 10 to the negative 2 is, you would take a 1 and you'd move its decimal point backwards instead of forwards. So 1 would be here, and then 2 would be here. So you put a 0 here. So that, and another way of writing that would be 1 over 100, right? I know you guys are probably looking at this like, yeah, we know about tens and all of this. But if this isn't starting to put it into perspective of what pH really is telling us, I feel like this is sort of breaking it down now. So when I say a pH of 14, what I'm really saying is a pH means the negative log of the hydrogen concentration. So we're really actually talking about a lower hydrogen concentration because we're talking about 10 to the negative 14. That's the concentration of the hydrogen ions. If I say pH of 14, our hydrogen ion concentration is 10 to the negative 14. That's literally what it means. Okay. So is 10 to the negative 14 a smaller number? Literally just 10 to the negative 14. Is that a smaller number than 10 to the negative 2? Hell yeah, it's a smaller number. Look at it. They wrote it out here for us. This is 10 to the negative 14. Is that smaller than this number? Heck yeah. How much money would you rather have? 0.01 or 0.0000000? Right. So now we're starting to understand. So that's what those numbers really mean. It's 10 to the negative of that number. So that's what pH really is. If I say something's at a pH of 2, it's 10 to the negative 2 of hydrogen ion concentration. That's why it doesn't go below 0. Because what's 10 to the 0 power? Anybody know? 1. Okay, go. Lower than that. Okay, that's like it. That's the maximum. That's pure hydrogen ion. That's what that is. Okay. So that's our scale, and that's how it works. Does that make sense to everybody? Anybody need me to explain it differently or anything like that? We good with pH scale? I know that you guys understand that less than 7 is acid and greater than 7 is base. I'm for sure going to be talking about that, and there probably will be a question about that on the test, right? Like, okay, your patient's urine sample was a 8.4. Is that considered acid or base on the scale compared to water? And then we base, right? Because it's a higher than 7. However, if I were to say which one has the most hydrogen ions in it, a pH of 5 or a pH of 10, which one has the most hydrogen ions? You guys know? 5, right? Okay, so now we're getting it a little bit better how to apply that because both of those types of questions will be on the test. One of each of them. That's not like the whole test is that. Okay. I'm not asking you to do the math. I'm just asking you to recognize where it would fall on that scale and what is actually going on there. Because now we can start seeing if your body pH is supposed to be 7.35 to 7.45-ish, that if we start falling down below that, what we're really talking about at a pH of like 6, what we're really talking about is too many hydrogen ions. And then those too many hydrogen ions, they're hanging out with actual positive charges. They're not polar. They're actually positively charged. They don't have their electron. They don't have any more Barbie at all anymore. Somebody completely took their Barbie away from them. They want to at least be sharing it with somebody, or at least be associating with somebody that has a Barbie, because they are Barbieless. So they're coming out with their H plus little attitude. I have no Barbie. I ain't never had a Barbie. Great for you that you have a Barbie. I'm going to hang out with you and make it look like I got a Barbie. Sort of situation, right? So they're looking for those negatively charged oxygen ions. And they're happy to come in there in the middle of your DNA and interfere with the other hydrogen that is only polar. It's sharing a Barbie with somebody. It's just not getting as much time as it would like. But it's happy to come in there and interfere with that and break up your DNA bonds. That's how some of our cleaning agents actually work, is by interfering with those hydrogen bonds. pH has a big part of that. Heat can also break up those attractions, of course. We're going to talk about that as we go through the course. So while we're keeping these concepts of how hydrogen bond works, why it works, how pH has a part in that, now we're starting to get why that's important and why we're driving it home so hard, right? Cool. We know what an organic molecule is. We know it's based in carbon. But if it's just got carbon and other inorganic, like other oxygen or something like that, like carbon dioxide. Is carbon dioxide an organic molecule? I mean, it literally says in here. No. Right? It's not organic. You have to have some kind of hydrogens in there. for it to be organic. So just remember that. Carbon-based compounds are going to be in our organic. You have to have carbon for sure. But if you're not having hydrogen to support it, it doesn't really count as organic in our book. Okay. Carbon is not the end all be all. They got to have hydrogen supporting it. And that carbon hydrogen association, a hydrogen has never been happier than it is with a carbon. It is so happy there because it's finally sharing that RB equally. Finally. Man is oxygenaholic. And our biggest players probably in biology are going to be your carbons, hydrogens, and oxygens. And we've got nitrogens coming in. We got phosphorus. And we got some sulfur coming into play too. But those carbons, hydrogens, and oxygens make up the biggest part of you and what makes you you. Those oxygens want to hog those electrons. Those carbons don't. They want to play fair. And that's going to dictate basically what the structure is of all the molecules in your body. So now we're starting to get it. So here's our carbon. It likes to make 4 bonds. It's very, very happy with that. Even when it's bound to an oxygen, those oxygens aren't really electronegative enough to make a carbon unhappy. Maybe a little bit if we're getting super like physical chemistry level of stuff. Okay. Sorry. If you're going to med school later, you're going to get into that in pharmacology and things like that. But you guys, we're not going to worry about that too much. Think of just always carbon, always want to play equal, and the things that are bound to it also getting to play equal because they're bound to carbon. The great mediator are carbon. Okay, that's what we love about a carbon, that nice stability that it gives us. So now we can get to talking about our macromolecules in biochemistry. Biochemistry is the chemistry of life. Okay, our 4 big groups: carbohydrates, lipids. You can think of them as fats. That's fine. We don't need to get super specific in this course. Okay. Carbohydrates, lipids, proteins, nucleic acids. Those are our big 4. We're going to go through each one. And each of these guys are considered our biological macromolecules. And a macromolecule is something that's built up of smaller parts. The smaller parts are called monomers. Those monomers come together to make polymers. For carbohydrates, our monomers are called saccharides. They're also just sugars. Just a nice fancy word for sugar. The saccharide. So monosaccharide is one single sugar. And then you have disaccharides, di being 2. Polysaccharide, just a whole bunch of them now, more than 2. Okay. You can have sugars, basically all the sugars that we have, and like we name them, because you got to have special names for everything. They all end in ose. So glucose, sucrose, fructose, lactose, all of those guys. You hear an ose at the end, you should automatically in your brain just be automatically hearing that's a sugar. So you guys know what sugar is in DNA and RNA. So let's start with that. Ribose. Okay, ribose. So ribonucleic acid, which we're going to get to in a moment. Ribose is the sugar that's in RNA. The sugar that's in DNA, deoxyribose. So I'm going to tell you, ribose is a 5-carbon sugar. We're probably going to need to know that, just FYI, because especially when we get into chapter 6, when we start talking about DNA and RNA and their functions in the body, it's going to help a lot because these are pentoses. They're 5-carbon sugars, and it's different than our hexoses like glucose, which is a 6-carbon sugar. Glucose, and if you have taken physiology, then you understand how important glucose is and understanding how your body makes energy to support all the processes in your cells. Glucose is our king of our biological sugars, okay? Our king of our 5-carbon sugars is going to be ribose. And I picked ribose, even though we like to think of DNA as being so all important and everything in your genetics. But what is deoxyribose except for ribose without an OH group, oxy? So how many carbons do you guys think deoxyribose has? 5. If I tell you ribose has 5, and the name of that chemical is deoxyribose, how many carbons have we lost in that? So it's a 5-carbon sugar. A 5-carbon sugar, okay? Ribose is a 5-carbon sugar. Deoxyribose is just ribose without one of the OH groups. Is there a carbon in an OH group? It's oxygen and hydrogen. It's not carbon, okay? All right, we'll talk about that more. I'm not gonna make you pick these guys out of lineups. Here's some pictures. That's pretty much what this is, okay? So you guys know what sugars look like. You know, they have this nice, pretty hexagonal ring. If they're 5-carbon, they have a nice, pretty pentagon type of a ring. As long as you can kind of recognize them in these shapes, because we're going to be showing pictures of a lot of these chemical groups. If you were taking biochem, I would absolutely expect you to know where all these carbons are, where these OH groups are, and be able to draw all of these and their orientations and everything like that. I had to learn that. But I don't care for this for my course. For my course. I can't guarantee you that in Dr. Shearer's course, okay? Sorry. So, anyways, moving on. So that's our sugars, the oses. They are, monomers for those guys are our saccharides or our sugars, the carbohydrates. And moving on to the lipids. These guys aren't going to have a true monomer for our purposes, okay? But if you're going to have a take-home message for our lipids, it's that generally speaking, they have some at least very large hydrophobic acid, or hydrophobic, aspect to them. So these guys, hydrophobic. If you hear lipid, fat, phospholipid, wax, all of these guys that have some fat, lipid, these guys have long hydrocarbon tails to them, at least at some part of them. That's going to give them a hydrophobic characteristic. That's the big take-home message with these guys, these hydrocarbon chains. We have some groups that kind of fall into this category, like our triglycerides. I'm really not going to get too much in the chemistry of these, but this is kind of what they would look like. We have a glycerol group, and the OH here is just to help attach our little fatty acid chains onto here. And so we have 3 fatty acid chains hooked onto a glycerol molecule, and that's why it's called a triglyceride. You're never going to have to draw one. You're never going to have to recognize one, but here I'm breaking it down for you. And most I'm going to ask you about this is like maybe what category it falls into, the lipid category. So does it have a hydrophobic aspect to it? Yes, because we just said that. And why? Why does it have hydrophobic aspect? Hydrocarbon tail. Look at that. Beautiful. Super stable. I wish I was that stable. All right. Fatty acids. So these can be saturated or unsaturated. I think we had a picture. Let's go back to this other picture. You see how these guys have double bonds here? We call that unsaturated, because if you were to switch that into a single bond, you'd have another hydrogen hanging out, right? To keep the 4 bonds on your carbon. So yeah, this carbon has 4 bonds, just one of them's 2. If we switch this to a single bond instead, you'd have to have another hydrogen coming on. So we would call that saturated. And that's why this one's saturated. You see how it has all the carbons are just single bond. We have nice hydrogens coming off of them. That's literally what saturated means. So if it's unsaturated, it means you have some double bonds in there. Why is unsaturated more healthy for you? The unsaturated guys are less likely to solidify and stuff in your arteries and things like that, because they have those little kinks in them. It just makes them a little more fluid. There's other reasons too, but that's like the baby version. Okay. Yeah. All right. So then moving on to our phospholipids, we already talked about this guy. We kind of drew a really bad picture of it and all that, but here's a nice good picture of it. These guys have a nice phosphate head, and then they have 2 hydrocarbon chains coming off of them. Nice fatty acid hydrocarbon chain. So we have a hydrophilic, water loving, and a hydrophobic, water hating or water fearing. These guys don't love water. Okay. So we are amphipathic here, right? That's what we would call that. So these are great because these polar heads that we have here in our little circle is what we're having in our little diagram down here. Our little polar heads, because they got that charge, they're ready to party with water. They're happy to party with water because they've got that charge going on. These tails, they do not want to party with water. They don't want to party with water just as much as your olive oil doesn't want to party with water. They hate it. They're against this, right? They are, don't get a little political, but they're the boomers at the party, okay? They don't want to have anything to do with that party. So they are going to do everything they can to hoard themselves away from the water party. So what they'll do is they'll stick together and they'll put the heads on the outside. Does anybody remember what this structure here is called that we're forming right here, where we have like these polar heads on the outside and we're starting to get the tails on the inside? Does anybody know what that like circular kind of structure we're forming here is called? Anybody remember? Micelle. You guys remember that? Might have come up somewhere. You guys ever heard of micellar water? So this is water, ladies probably have, right? Because we know it's good for removing our makeup and stuff like that. Why? Because it dissolves fats better. Because we're putting in an environment there that's a little bit more friendly to fats, which means it's also more friendly to your lipid bilayer, because that's what the membrane on your skin is a lipid bilayer, which we show in the picture right below. Lipid bilayer, we've got these charged heads ready to party with water on the outside, and we've got the tails hiding out on the inside, our water-fearing parts. We've got 2 layers of that facing each other. And that structure, 2 layers, the hydrophobic on the inside and the heads, polar heads of the phosphates on the outside, create an entire barrier around your entire cell. And now, if that is the weirdest chemical composition that anything could want to pass through for all time, you've got weird, heavy charge on the outside, you've got hydrophobic on the inside. What molecule can pass through that naturally? Really, there's only one that can comfortably do that reliably, and that's water. And so now you can have water going in and out of the cell freely, but you're keeping everything that you need in the cell inside and everything that you don't want in the cell outside. And now you can control that. And if we want to help things get in and out better and maybe move, nutrients and things like that in and out of the cell, now we can put channels in that with our proteins and things like that. So we can control what moves in and out of the cells. But that lipid bilayer, that phospholipid bilayer, that's the king. That's what makes up pretty much every cell's membrane ever, except for archaea, because they're weird. Okay, they have like the weirdest. It's not this. Okay, they're on like some other, like oxanes. It's like weird. I don't even know. Okay, so we're not talking about the archaea. We're talking about bacteria, and we're talking about eukaryote. Okay, you don't have to learn about the archaea. They're membranes, anyways. All right, so moving on. How do we give structure to our cells? So for our cells, and I'm sure you guys learned this in like basic middle school biology or whatever. We don't have cell walls, right, on our cells like plants do. That's why they have such a rigid structure, why they get bark and all this stuff. They can hold themselves up with their cells. They get a cell wall. Bacteria also have a cell wall. It's not made out of cellulose like plants, it's made out of peptidoglycan. We'll talk about that in chapter 4. But we don't have a cell wall to support our cell structure. So what we do is we put these other more sturdy lipids into our lipid bilayer to give it some more structure and shape. So now we can have shape and structure in our cells, right? And that molecule that we use for that is cholesterol. So look at this. You see this OH group all the way up here, the one lonely oxygen in all of this. That helps it kind of snake in there with those polar heads. So it can kind of start to associate. But all that CHs and all that down there, that's what makes it hydrophobic. So now we can get in with those hydrophobic lipids that are in the middle of all that. So it can kind of get stuck in there. And all those nice, beautiful rings, again, you don't have to recognize it in the picture, maybe in Dr. Shearer's class, okay? Not in my class. But you can see that the rings would give it structure and stability. So we got this nice, big, structured molecule that gives stability to our membranes. And when you get this floating around in your bloodstream, what did we say about lipids? What property do they have? They are hydrophobic, right? They're water-fearing because of those hydrocarbons, right? Cholesterol is one of these. All the sterols are. All the steroid-based molecules are. So we've got this nice, good, structured ring structure. They're all hydrocarbons, so it's water-fearing. So what's going to happen to these guys when we put them in your bloodstream? What's your blood mostly? Water. And that has polarity. So these guys don't want to party with that. They don't. That's not their scene. So they're going to stick together and they congeal up in your nice little blood vessels, get stuck in your capillaries and your nice little arteries and veins, especially the nice little delicate tiny ones that feed the oxygen to your heart muscle, your coronary arteries, and get nice and clogged in there where you need oxygen the most and cause some serious problems, right? So now we're getting into that medical side of understanding. Now we know why cholesterol congeals the way it does in your blood vessels. Because it's hydrophobic, it doesn't want to party with the water in your blood. But it has its uses. It gives structure to your cells. We need it. What are you going to do, right? Too much of a good thing. So all of our steroids kind of have that similar sort of function. They're either going to be involved in making hormones, which we're not getting into in this class. That really is a Dr. Shearer thing, okay? But, or it gives stability to our membranes. For like animals, we have cholesterol to give stability. Fungi, they have ergosterol. Okay. You guys heard of that? What is it? That rye? It's like rye. I really feel like it is rye, and it gets a fungus, and it's like ergo something. I can't remember what the name of it is, but it makes you hallucinate and have like crazy hallucinations. Like a whole town in like Canada, they had an outbreak of this. See, you're nodding your head back there. You know what I'm talking about? Okay. So there's this fungus that grows on rye, and there was so much of this fungus, and these people were like growing rye in their fields or whatever, and it got contaminated with this fungus. And they didn't really think about it. It wasn't bad for you really to like have contaminate in your food, but it would get in the air and make you hallucinate. And like the CIA literally investigated into this and like how they could have maybe have whole towns be affected by this and investigate. I'm not kidding. This is like some MK Ultra stuff that's actually happened. Look into it. But that ergo stuff, every time I read that word, I always think of that. Ergot, right? I think that's right. Yeah. Anyways, the more you know, folks. All right. So then we also have waxes. Obviously, if you're waxing your car, we usually do that to help the water that might get on it and the dirt that's contained in that water just bead up and just roll off of your car. It's hydrophobic. We're putting a hydrophobic thing on your car so the water doesn't stick and dry off on your car, right? So we know wax is hydrophobic. We're going to talk about a particular wax called mycolic acid in chapter 4 that is very, very important for certain pathogens like tuberculosis. And we'll get into that later. All right, so proteins are our next guys. These guys are probably your workhorse in your body, in your cells, okay? We'll talk about a little bit about what their functions are and how they function, what they do for your body, like all throughout the semester. We'll get to them. We're going to talk about their structure and stuff right now, though. So proteins, the shapers of life, they have a monomer. We're back into the monomer thing. Like I said, you didn't need to know monomers for the fats. What were the monomers for the carbohydrates? Saccharides, sugars, right? Don't care about monomers for your fats. I really don't. But when we get into our proteins, we're back into monomers. What's the monomer for a protein? Amino acid. So proteins are built up of repeated amino acid units. I'm going to show you what an amino acid is. We know there are 22 that exist in nature. There's 20 main ones that we'll see mostly in like animals and stuff. Here's our list of amino acids. I'm not going to make you memorize the list. Sure as hell, you're going to have to learn this if you take biochem, right? You're going to have to not only know them, but be able to draw them all. But we don't even know that. But just so you know, you've probably heard of some of these guys. And these guys are amino acids. You can see there was about the R group, and I'll remind you what that is here in a moment. But that is basically what determines whether you are alanine versus cysteine versus glutamine or whatever else. Okay. So that R group determines what you are. And that R group might have a characteristic of being polar or non-polar, or might have a full-on committed charge where it's donated or it's gained an electron, right? Like our ions do. So these aspects, now you can start to see how important those hydrogen bonds are going to be in creating our structures of our proteins, as we have thousands of different amino acids, and how those R groups interact with the guys immediately next to them versus the guys on the other side of our polypeptide ribbon. All right, so here we go. Here's an amino acid. In the middle, you always have a alpha carbon. On each side of the carbon, you have your amino and you have your acid. Amino is the NH2. That's your amino. The acid in the amino acid is carboxylic acid, or CO. You could think of it as CO2H or whatever, but it's like there's two oxygens, one with an OH. Okay. That's basically what that is. And that's the basis for all amino acids, okay? And then if you want to hook this one to another one, you would just basically paste them together side by side by side. The amino to the next carboxylic acid group, and the amino to the next carboxylic acid group. Draw it for you. I wanted you to see it, but I'm not gonna. So I'm not gonna make you draw it. But anyways, when you bind an amino acid to another amino acid, that carboxylic group binding to the amino group of the next guy, that bond that occurs between them is called a peptide bond. We talk about peptide bonds a lot. We'll be talking about peptides a lot, polypeptides, and proteins a lot. Okay? So it's kind of important to get that. And when we start talking about amino acids and the different amino acids, this R group is the one that's blocked out in the different colors on here. That's what determines whether you're tyrosine versus phenylalanine and whatever. Cool. So peptide, we talk about peptide bond. That's the bond between the amino and the carboxylic acid of the next amino acid. All right. The peptide itself means that we have a few amino acids linked together. That's a peptide. They're short little chains of amino acids linked together. A polypeptide, now we're exceeding 20. Now we go up to thousands, but you might only still qualify as a polypeptide, because a protein is once we get to that level that it has a function. And some of these guys don't function as just one single polypeptide. We have to have multiple polypeptides come together to make a functional protein. We're going to talk about going into the structure of proteins. Our primary structure is just how you would spell out your protein. So alanine, phenylalanine, alanine, phenylalanine, tyrosine, alanine, phenylalanine, phenylalanine, tyrosine, whatever it spells out to be with whatever the amino acid is next in the sequence. That is all determined by your genetic code. We're going to talk about how that plays out in chapter 6. Don't lean too much into that right now because we're going to talk about the specifics of how that works in chapter 6 when we get into genetics. Okay, but all of your amino acids in a row, all dictated by your genetic sequence. Then we're spelled out. And by the way, your genes, they all code for proteins or things that help build the proteins or control the building of the proteins. They don't code for sugars. They don't code for fats. Nope. They code for proteins. And then those proteins might be signaled to put together fats or put together sugars or whatever, based on other signals they're getting from other proteins or other molecules in the cell. Your genes don't code for a damn sugar or a damn fat or anything like that. They only code for proteins. Proteins are king. You have about 20,000 genes in your body, okay? You can make 8.5 billion different antibody proteins. Typically, you need a single gene for a single protein. Talk about how that works later. It is so cool. But anyways, proteins are king in how your cells and how your body are going to function. Basically dictate everything. And hey, let's say your body codes for a protein like keratin, that's your waterproofing protein for your skin. How are you getting that to the surface of your skin? Well, you made your little signals and your body told you to make keratin. There's a whole slew of things that goes on with that. But let's say you make your keratin and all of the machinery that leads to making the protein made it for you. How do you get it out of the structure where that occurs, which is your endoplasmic reticulum? How do you move it out of that endoplasmic reticulum and put it on the surface of your cells or the surface of your skin? How do you get it there? Does it just appear there? Does it just migrate there because of diffusion? No. How does it get there? Vesicles inside of the cell are going to carry it out to the surface of the cell. How do the vesicles move? They are literally carried by proteins that literally carry the vesicles on their back and walk them along your cytoskeleton to, I'm not joking, this is how your cells work. Literally everything is governed and moved and everything happens thanks to proteins. How are you driving that protein carrying that vesicle on its back to the other side of your cell? You need energy. We'll talk about that in a little bit too. Okay. So anyways, you built your protein, you spelled it all out. Alanine, phenylalanine, tyrosine, alanine, phenylalanine, tyrosine, cytosine, whatever it is, right? You spelled out your protein. Now you made your ribbon. So that's our ribbon. That's our starting point. That's your primary structure. Your secondary structure. We got a whole lot of charged guys in this part of it, of our ribbon, and a whole lot of hydrophobic guys in this section. Maybe a mixture over here. Well, those guys are next to each other, crammed up right next to each other. They're going to start moving away or near the guy that's right next to each other and adapting to the chemistry immediately next to them. That's going to create our secondary structure. The R groups immediately next to each other, creating all this movement and secondary structure, okay? There's two main structures that can evolve from that, styles of structures, if you will. Alpha helices and beta pleated sheets. An alpha helices means that ribbon is starting to curl on itself, just like a helix, okay? A beta pleated sheet means we have lots of straightaways that are going to fold back and fold back. This is a beta pleated sheet. Doesn't matter what the chemistry is that causes that, okay? But you just need to know what they are, and that those are part of the secondary structure. All right, so this is like you took your ribbon and you curled it with your scissors while you're making your package, okay? Right. Tertiary structure. Now we're folding our ribbon, tying it in a bow. We're taking that one lonely ribbon that we curled in our secondary structure, and now the things that were far away over here, these alpha helices over here, they're just coated on the outside with these charged molecules, and on the inside hiding all these hydrophobic guys. Well, maybe this whole section over here, it has a way more hydrophobic that wants to hide on the inside of the whole structure. So we're just going to kind of have because we're in water, have all of our charged guys more on the outside and our hydrophobic guys more on the inside. So now start getting big picture folds. And we'll even create covalent bonds across the way from each other with like what we call disulfide bonds, things like that. That's our tertiary structure, how the big picture stuff starts to fold. Think of it as tying the bow. All right, so we curled our ribbon, we tied the bow. That's our tertiary structure, but we're not done. Your package that you're wrapping looks like crap with just the one bow on it. Sorry, but that's just facts. I'm just being honest to you. So put a whole bunch of bows together to make it a nice big old bow. Now we have a nice, pretty, functional package that's ready to ship out to wherever you want it to go. That functional thing that we just made with multiple bows put all together to make it look nice and big, that's our quaternary. Most proteins that have a function in your body, they have more than one ribbon. They have more than one polypeptide. And they come and they work together in order to make a functional protein. These proteins have functions like enzymes that help catalyze reactions. A catalyst is something that helps a reaction happen without costing energy as much. We'll say without costing as much energy as it would without its help. And the enzyme, which is the organic molecule that helps drive these reactions with less energy, is not used up in the process. We can reuse it over and over. It's not permanently altered. Might be temporarily, but it can just revert back to the original and be reused and reused over and over again. That's enzymes. They help our reactions go faster, and we'll learn about them in chapter 10. In chapter 16, we will learn about antibodies. These are proteins that are made by your body that help target foreign things. We'll go and say, you are not self. I've been trained for this moment. You are not self. I recognize specifically you as being foreign, and I bind to you, and now the body can target it for all sorts of immune reactions. We'll talk about that in chapter 16. So the proteins that are essential. Now we have to have our bows tied in specific ways, just like we had to have our double helix have our hydrogen bonds in its middle to hold it together. These proteins often rely on hydrogen bonds across the way to help hold that bow structure together. We interrupt that with too many H pluses, too few H pluses, heat or other chemicals interfering with that. Now the structure doesn't want to hold. The bow doesn't want to stay tied. I have these boots, y'all, that when I tie the bow on them, no matter what I do, that bow comes untied, okay? Think of that as denaturation. The natural state where it wants to be, that bow tie where it's actually going to be functional and holding my boots on, that's the native state. Some sort of heat from me walking around might be what's breaking up its ability to hold its bow shape. And now it's no longer functional. Denaturation. Okay. All right, finally we're into the nucleic acids and then we'll be done. We have DNA and RNA. DNA, of course, our genetic information. RNA is going to be our working copy of whatever the gene is that you're dealing with. We'll talk a lot about that in chapter 16. These guys consist of monomers called nucleic acids or nucleotides. The nucleotides are the specific little structures. Nucleotides consist of a sugar, and then there are pentoses. This is definitely a pentagon, okay? And then I know, it's very sad. And then they're hooked to a phosphate. We already know that that phosphate has a ton of oxygens on it, so it's going to have that charge to it. And then off of that sugar, we also have branching off of it a nitrogenous base of some kind. They've got, like, look at them, y'all. Look at these guys. And I'm not going to have you recognize the shapes. I'm not going to have you remember that they're purines or pyrimidines, not in this class, but you do need to know that they're A, T, C, G. A is adenine, T is thymine, C is cytosine, and G is guanine. That's not going to be tested on unit 1, but we are going to test that on unit 2, chapter 6, the first chapter of that. We'll come back to those and have to know them. But that's the structure of a nucleotide, but you do need to know the general structure of a nucleotide and what makes up DNA and RNA, okay? So what's the difference between DNA and RNA? That sugar in RNA is ribose. Take away one of the oxygens on it, and you get deoxyribose. That's the difference, okay? Also, the T is only in DNA. It's a U that shows it on the other side. It's just a different version of it for RNA. And that helps us kind of have that separation and separation of function as well. Not having you know about purines and pyrimidines, you're welcome. All right. And this is how it would look like. We have a nice little breakdown of like the DNA backbone, the backbone being sugar alternating with phosphate. And in the middle, we have our bases, the A's and the T's and the C's and the G's all meeting up. A always binds across from T. Think of those guys as having, like, they have very harsh lines, right? An A and a T, very harsh, pointy lines. G's and C's have the nice roundness. The G's and C's bind to each other. The harsh A's and T's bind to each other, okay? All right, RNA, again, they're our messenger. And they also comprise the things that are going to be involved in making the proteins. We'll talk about that in chapter 6. Finally, we get to ATP. This is our energy molecule. Your cells rely so heavily on ATP, which is adenosine triphosphate. It's basically ribose, adenine, which is the A from, like, this is why it's in the nucleic acid group. It's ribose, it's the A, nitrogenous base, and triphosphate, 3 phosphates. Now, these are hella negatively charged. We already talked about the PO4s, having all those oxygens, stealing all those Barbies, being all greedy with their electrons. These negative guys don't want to be near negative guys, near negative guys, near negative guys. And they are forced by covalent bonds to share with one another. They hate it. And all they want to do is get cleaved off. And when you cleave it off, they are ready to party. And they release all this energy. And your cells can use that energy to drive all the processes that it needs to carry vesicles on their back across to your cell, or drive making new DNA, or drive making more proteins, or whatever it is your cells need to do. In fact, so essential to your survival, ATP. If I suffocate you with a pillow right now you might think I'm suffocating because I'm not getting oxygen. Exactly. What do you think you need oxygen for? To make ATP. That is the only reason why you breathe in oxygen, is to make ATP. And you run out so fast, within minutes. A multicellular creature like yourself, within minutes you'll die because you don't have enough ATP. ATP is it. That's the reason. We're going to talk about aerobic respiration and why you need oxygen to drive that process later on. Spoiler alert, the carbon dioxide that you breathe out, the CO2 you breathe out, the O2 in that CO2 doesn't come from the oxygen you breathe in. Those of you who are in physiology, you know where it comes from. The CO2 that you exhale out of your body as gas came from the food you ate. It didn't touch that oxygen you breathed in. Cool stuff. We're going to talk about that in chapter 10. And that, to me, I think aerobic respiration is one of the coolest things you'll ever learn in science. I think it is just so cool once you finally understand why you breathe in oxygen and exhale carbon dioxide and why you suffocate within minutes. So cool. How that's all driven by that, and why you need ATP for energy, for all those proteins that are doing all this work in your body. And it starts to come full circle. Why you need to keep your pH stable, because you have to keep all those proteins functioning. All of that's going to tie together. So that's it. We